Hi, good afternoon. I think we can get started. This is Judy Lieberman, and I'm very happy to host Shijeng Su from Sun Yat-sen Memorial Hospital. Shijeng is a breast cancer researcher at Sun Yat-sen and a surgeon, an MD PhD, who's done really innovative work studying the tumor microenvironment, especially the more innate side of tumor immunity. He's always coming up with completely unexpected and original stories that link new cells, new pathways, new molecules, non-coding RNAs, receptors that nobody has ever heard of, to the immune response to breast cancers. And today he's a professor at Sun Yat-sen. He's actually the director of the Breast Cancer Research Center there. He told me that they've just set up a hospital at Sun Yat-sen that's only devoted to breast cancer. And today he's going to give us a talk on beacon towers of the Great Wall, new nucleotide sensing membrane proteins, and the inflammatory microenvironment. On the first slide, you can see the website of the Connect Science seminar series, and you can go there and find out about the amazing cancer-related seminars that we host. And if you have questions, please enter them in the Q&A at the bottom right of your screen, and we'll ask them after he finishes talk. Shijeng.
Thank you very much, Judy, for your kind invitations and nice introductions. It's my profound honor to be here to address the major interests of my lab. That is how nucleotide sensing proteins on the plasma membrane or the organelle membrane regulates the fate of immune cells in the inflammation-associated disease. The innate immune systems recognize a broad range of molecular structures that are conserved amongst different microorganisms. All those pathogens are key involvement. Nucleic acids are an intrinsic part of their structures. Therefore, recognitions of nucleic acids are important for the immune surveillance. The nucleotide sensors can fall in two categories, the DNA and RNA sensors. They can also be divided into two groups according to their locations. The first locates in the endosome, such as toll-like receptor 3. The other ones locate in the cytoplasm, such as regard cGAS-STINs. Please notice all of these sensors detect intracellular nucleic acid. So how about the extracellular acid? Is there any sensors for the extracellular nucleic acid? Moreover, the downstream pathways of this nucleotide sensor are overlapped and redundant. It consists of NF-kappa B and IIF pathways. So, is there any other downstream signaling pathways beyond the inflammatory pathways? Emergent studies show that the extracellular nucleotides are also important for the inflammation-associated disease. For example, neutrophil extracellular tract, NETs, are large red light structure released from the neutrophils. It consists of the condensed components and neutrophil proteins. So, how NETs are generated? The NETs can be stimulated by the microorganisms or the endogenous stimuli, such as immune complex. The stimuli induce the burst of ROS. The ROS can activate PAD4, which is an enzyme that converts arginine to citrulline on the histones. As a result, nucleus components decondense into the cytoplasm. The neutrophil proteins can be released from the granules and coated with the DNA filament. This leads to the further unfold of the chromatin and expels into the extracellular space through the ruptured plasma membrane. Please notice the pathway and compositions of the net varies dependent on different stimuli. It was called NET because it first reports to help neutrophil to check the pathogens. However, recent studies show that NETs also play an important role in non-infectious disease, such as cancers. In the murine model of infections using LPS injections, cecal ligation and punctures, ischemias, and reperfusions, it has been shown that the NET formation is important for the metastasis to liver and lungs. However, the question remains, what is the role of NETs in more physiological relevant models, and whether the effects of NETs are different in various metastatic organs? More importantly, what is the clinical significance of NETs in cancer patients? Moreover, how NETs interact with cells is even more poorly understood. It has been shown that the granule proteins coated with the DNA filament can await the tumor cell and promote tumor metastasis. However, what is the roles of the DNA in regulating the interactions between NETs and cells? Because administration of DNAs, which degrades the NET-associated DNAs, can significantly reduce the NET-associated metastasis. This data suggests that the DNA component is also important for NET functions. So how DNA interacting signals are transmitted from the extracellular space into cells? To address these important questions, I collaborate with Dr. Soames to investigate the clinical significance of NETs. We conduct the immunofluorescence staining for the MPO and the circulating histone 3. Both of them are specific markers for NETs. The NETs were quantified as the percentage of the fields positive for both these two markers by the automated image analysis. Our data show that field NETs can be detected in the primary tumors. In the metastatic lesions of brain, lungs, and bones, the NETs can be detected in few cases. By comparisons, liver metastasis exhibits abundant NET infiltrations. It has been shown that the MPOs DNA from NETs can be found in the blood of the patients with autoimmune vasculitis. So we ask whether they can be also detected in the blood of cancer patients. Therefore, we coat the microtiter plate with the anti-MPO antibody as a capturing antibody and incubate the patient serums with the peroxidase labeled anti-DNA antibodies. Then we apply the substrate of peroxidase, and the concentrations of MPO DNA levels was quantified by the optical densities emitted from the peroxidase and read by the microplate reader. Our data show that the levels of serum MPO DNAs increase in the patient with the liver metastasis compared to the patient without metastasis or metastasis to other organs. Moreover, the serum MPO DNA is an independent variable associated with the long-term liver's metastasis-free survivals, but not with the metastasis to other organs. The ROC areas is 0.863, suggesting that the serum MPO DNA is a good biomarker to predict the future liver metastasis in early breast cancer patients. To investigate whether the mouse model can recapitulate the clinical findings, we ingest the human breast cancer cell in non-skin mice fat pad. We assert abundant NET formations in the liver tissues, but only low levels in the lung tissues. More interestingly, we assert NETosis as early as 15 days after the injection whilst that metastasis is only detected after 30 days by the human genes expressions. This data suggests that the NET can be released from the neutrophil in the premetastatic niche of livers. As we previously mentioned, that the PAD force is crucial for the formation of NET. Therefore, we use the PAD4 knockout mice. We ingest that the mouse breast cancer cells in the spleen of syngeneic mice deficient in PAD4. We observe that both netosis in the livers and the liver metastasis are greatly reduced. Given the NETs are presented in both blood and liver metastasis and closely associated with the liver metastasis, we ask whether the NET can serve as a chemotherapy factors. Therefore, we develop a protocol to extract the DNA from NETs. The PNA is a useful chemical which leads to first identifications of netosis. Therefore, we apply PNAs to the primary human neutrophils to induce the netosis. After removal of the supernatants, we connect the adhered nest by pipetting with PBS. After centrifugation, the cell-free net DNA potent complex are connected, and the DNA are isolated by the microelude DNA cleanup kit. As I previously mentioned, that the walls play important roles in the formation of NET. AOHDG is one of most predominant form of free radicals induced oxidized lesions. Our data showed that the AOHDG level increase in NETs compared to the unstimulated neutrophils. The TRES1 is a 3' to 5' extranuclease, which then degrades both single-strand and double-strand DNAs. But the oxidized DNA is resistant to the TRES1. We observed that the NET DNAs that we isolated from the PN8-treated neutrophils is resistant in terms of dose and time compared to ones isolated from the untreated neutrophils. Moreover, we place the NET DNAs on the lower chambers and tumor cell on the upper chamber and observe that the NET DNAs can attract the tumor cell in dose-dependent manners, which can be abrogated by the DNAs. The mule-chambered assay also showed that the cell efficiently migrates toward higher gradients of DNAs, of NET DNAs. Given the NET DNA can assert the function of chemotaxis factors, we ask whether there is a receptor on the cell surface which detects the NET DNAs. To isolate the putative NET sensor on the cell surface with labels that NET DNAs isolates, as we previously mentioned, by octants. And then we applied sulfur NHS by octants to the suspension of tumor cells. NHS can bind to the primary amines on the cell's surface. Therefore, we lyse the cells and the bitinary cell membrane proteins were pulled down by the Wilding's agarose, which were later released by incubation with the IPS lysis buffers. And then we combined the NET DNAs and tumor membrane proteins, and then we tried this potent DNA complex by the Sheridan's magnetic spins. We tried potents at the size of about 22 kilodaltons. The mass spectrometers identified this potent as CD25 with little piece node functions. The DNA pull downs confers a specific bind of recombinant CD25 to NADH8, which can be competed by the enabled NADH8. The MSI assays show that the CD25 knockout can abrogate the potent and NADH8 complex formations. The apparent dissociation constant of recombinant CD25 to NADH8 is about 67 nanomolar, suggesting an efficient bindant. Moreover, the competitive experiment shows the CD25 preferentially binds to the AOHDG enriched DNA. Taken together, this data suggests that the CD25 binds to the oxidized NADH8. To investigate the roles of CD25 in vivo, we generate the CD25 knockout mice. The LTV PIMT mice is developed the memory tumors, the spontaneous memory tumors with considerable lung metastasis. We cause the CD25 knockout mice to the PIMT mice, and then we isolate the PIMT tumor cells from the CD25 mice and inject these cells in the spleens of the wild-type mice. We observe that the CD25 knockout significantly reduce the liver metastasis. It has been shown that the LPS can induce the NETosis in lungs and therefore aggravates the lung metastasis. Therefore, we treat the PIMT CD25 knockout mice with the LPS natural institutions and found that the CD25 knockout significantly reduced the LPS induced lung metastasis. To investigate how the CD25 bind with the NADH8, we use the transmembrane helix algorithms and predict CD25 has hydrophobic centers between residues 60s and 80s and two hydrophilic ends. To evaluate whether CD25 is transmembrane potent, we use a membrane fractions assays. We observe that the CD25 remains in the membrane fractions by treatment of high salts, strong denaturing, and high pH. By comparisons, CD25 solubilize in the supernatants by treatment of tritone. This pattern is similar to CCR4, which is also a transmembrane potent, but different from the ATP5B, which is a membrane-associated potent. So this data suggests that CD25 is a single transmembrane potent. The flow cytometries demonstrate that the CD25 with flat head and the N-terminus can be stained without cell permeabilization, whereas the one with flat heads at the C-terminus can be only stained when the cell was permeabilized. This data suggests that the CD25 has an extracellular N-terminus and an intracellular C-terminus, which was confirmed by the immune staining. The deletions of 40 amino acids on the N-terminus, but not on the C-terminus, can abrogate the binding of CD25 to the NADH8, suggesting that its N-terminus mediates these interactions. To pinpoint the specific sequence responsible for this effect, we compare the amino acid sequence of CD25 to the ones of several nucleotide binding potents. We observe that the amino acid 21 to 25 of CD25 was aligned with the DNA binding domains of HMGB1. Mutations of these domains abrogates the NADH8 bindings to the CD25 expressing cells. Moreover, we find that the ectopic expressions of wild-type CD25, but not the amino acid 21-25 mutants, rescues the actin polymerizations and directional migrations induced by NETs in the tumor cells with CD25 knockout. To explore the downstream signaling, we performed a pull-down assay using the lysis of HeLa cells transferred with his-test CD25. And the mass spectrometries identify ILK bias to CD25 under the net stimulations. Again, depictions of the intracellular C-terminus rather than the extracellular N-terminus abolish the bindings of ILKs to CD25. So how the ILKs transmit the signals of CD25 to the downstream pathway? Yes, we report the pigments binds to alpha PIX and provides a connection between the ILKs and the GDP bindings actin regulating proteins. The immune precipitation assays show that the ILKs recruit beta pigments rather than after pigments under the net stimulations. And CD25 knockouts abolish these bindings. Moreover, the net stimulations induce the GDP's bindings to the CDC42 and RET1. Also, this effect can be abrogated by knockouts of CDC25, ILKs, or the PAR1 silencing. Then we ask why the liver metastasis exhibit abundant net infiltrations. It's been reported that the complements can activate the formations of netosis. The overwhelming majorities of complement components are synthesized in the livers. Moreover, it's been reported that the complement can be activated in the plasmatic niche. Therefore, we ingest the tumor cell in the C3 locale mice and observe that both the netosis and the liver metastasis are greatly reduced. Taken together, our data suggests that the complements in the liver plasmatic niche induce the release of NETs from neutrophils. The NETs can serve as an chemotaxis factors, which attracts the circulating tumor cells from blood to livers. The DNA components of NET can bind to CD25, which is a sensor for the oxidize enriched DNA. CD25 is a single transmembrane protein on the plasma membrane. It binds to the net DNA with the amino acid 21 to 25, which is similar to the DNA binding domains of HMGB1. After the bindings, its C-terminus recruits ILKs. ILK initiates the beta pigment RET1 CDC42's cascades and therefore lead to the actin polarizations and the migration as well as adhesions. Then I would like to share another related stories. B cells are also an important stromal cell in tumor microenvironment. The previous study showed that B cells are pro-tumorals and immunosuppressive. However, the use of rituximab, the anti-CD20 antibodies, show no clinical benefits in solid tumors. This data suggests that both pro-tumorals and anti-tumorals B cells exist in the tumor microenvironment. All those several periods excellent studies identify a number of pro-tumorals B cell subsets. However, the phenotypes of anti-tumor B cell subsets remains unclear, and the factors which determines the opposite roles of B cells in tumors remain largely unknown. Previously, the systemic therapy is administered after the removal of tumors. So we do not know what happens to the tumor microenvironment during the treatment, as the measurable mass has already gone. The neoadjuvant chemotherapy is applied before the surgeries. Therefore, we can attain the pre-treatment biopsy samples and post-treatment resected sample from the same patients and investigate the dynamic change of tumor microenvironment during the treatments. Therefore, Yiwen, Qi, and Jianyou trying to dissect the dynamic change of tumor infiltrating B cell subsets before and after the chemotherapies. So they connect the pairs pre-treatment biopsy sample and post-treatment surgical research samples and performed the single-cell RNA sequencing. The single-cell RNA sequencing identified 7 different B-cell subsets in tumors around the chemotherapies. Consistent to previous reports, most of the B-cell subsets in treatment-naive states express high level of IL-10. Interestingly, a unique icosligand positive B-cell subset emerged after chemotherapy, which can be confirmed by the flow cytometry. To investigate the clinical importance of this B-cell subset, we performed the immunofluorescence staining. The automated imaging analysis of the entire slide sections show that the chemotherapies increase this B-cell subset infiltrations in tumors. Moreover, its infiltration is closely associated with improved response to chemotherapies and the better long-term survival. More interestingly, we found this icosligand positive B-cell predominantly located in the tertiary lymphoid organs, and these B-cells has closer contact with T cells compared to the icosligand negative counterparts. It has been reported that the tertiary lymphoid organs play important roles between the B cell and T cell interactions, suggesting these B cells may interact with the T cells to induce the anti-tumor immune response. Therefore, to investigate its roles in vivo, we use the JH knockout mice, which has no mature B cells. Consistent with previous reports, tumor growth in JH mice was significantly smaller than those in the wild-type mice and exhibit less Treg and more effective T cells in tumors. We confirm this finding by using the contact antibodies to depict B cells. But more interestingly, the B cell depletion mice has less chemosensitivity and effective Treg ratios. This data suggests that the tumor microenvironment exposed to chemotherapies can force an anti-tumor B cell subsets. The findings of icosligands in tumor-infiltrating B cells is surprising because of the previous report show that the icosligands in tumor microenvironment is mainly expressed in the myeloid cells. So we cross the CD90 crit mice to the icosligand FOS mice. In these mice, we observed that the icosligand level in the tumor-infiltrating B cell is significantly abrogated, whereas the DC from the spins remains unchanged. Using this B cell specific icosligand knockout mice, data show that the icosligand in B cells promotes the sensitivities to chemotherapies and induce an anti-tumor immunity by enhancing the CD8 T cells, Th1 T cells, and whilst reducing the FOS P3 Treg ratios. It has been well documented that the co-stimulated signal provided by the complement can enhance the B cell activations. Despite the importance of complement in B cells, immunities has been appreciated for more than 40 years. The roles of complement in the tumor-associated B cell remains elusive. Our single-cell RNA sequencing data show that the icosligand positive B cell subsets exhibit an elevated level of complement receptor 2, which is also termed as CD21. Therefore, using the C3 knockout mice, our data show that the C3 depletion significantly reduced the icosligand positive B cell abundance in tumors, as the complement receptor is not only expressed on B cells, but also on follicular DC and a subset of T cells. Therefore, the complement receptors can regulate that B cells at 2 levels. First, it can provide the direct signals to B cell. Second, the follicular DCs can deliver the C3 oxonized antigen to BCRs while the complement receptor 2. Therefore, we construct the B-cell specific 21 local mice and provide clear evidence that complement receptors on B-cell mediates the generation of ICOS ligand positive B-cells and its anti-tumoral activities. Clinically, we assert the biactive complement cleavage into human samples after the neoadjuvant chemotherapy. Moreover, the complement activation is closely associated with the tumor-infiltrating B cells. We also observe the complement is activated in the myeloid tumors. We assess the biactive fragment located in the proximities of blood vessels 3 days after the chemotherapy, and also located in the tumors, especially those with the terminal positivity 24 days after the chemotherapy. So how the complement is activated in chemotherapy? The phosphatidylserine sequence, PS, is a phospholipid exposed on the outer lipid of dead cells during the apoptosis. We observe that the accumulations of biactive complement fragments on the plasma membrane of the tumor cells, which are incubated in the human serums and treated with chemotherapy. The tumor cell exhibits the feature of dead cells with the caspase freeze activations and the part cleavage. The heat inactivations of human serums and neutralizations of PS can reduce the complement activations, whereas the tumor cell that remain unchanged. So this data suggests that the complement is activated by PS exposed on the immunogenic dead cells. The previous studies showed that the B-cell depletions can enhance the chemosensitivity in several mouse tumor models, such as CHAMP prostate cancer models. Surprisingly, we found the B-cell depletions reduce the chemosensitivity in multiple breast cancer models. This discrepancy is highly clinical relevant, as the B-cell infiltration is associated with both better and worse prognosis of patients who receive chemotherapy. Therefore, we screened the complement regulatory potence in the cell line endocytopedias and found that CD55 and complement inhibitory potence shows distinctive expression patterns among different breast cancer cell lines with different capacities to induce ICOS ligand positive B cells. The trans prostate cancer cells with a high expression of CD55 exhibits low ICOS ligand positive B cell infiltrations. However, the CD55 knockdowns significantly increase the ICOS ligand positive B cell infiltrations and enhance the chemosensitivities in these tumors. Taken together, our data suggests that the PS is exposed on the outer lipids of tumor cells during the immunogenic cell death induced by chemotherapy. The PS can activate the complement. The complement receptors on B cells mediates the generation of ICOS ligand B cell subset. This B cell subset induce an anti-tumor immunity by enhancing the T effector Treg ratio. CD55, which is an inhibitory complement potent, its expressions on the tumor cells determines the opposite roles of B cells in the chemosensitivity. In the previous next stories, our data show that the complement in the metastatic niche of liver promotes the tumor metastasis by inducing the netosis. In the B cell stories, our data show that the complements in the primary tumors convert the B cell from foes to friends. This data suggests that the complements in the tumor microenvironment is a double-edged sword, which is dependent on the concomitant factors. In the next story, we identified a nucleic acid interacting proteins on the plasma membrane. Within cells, multiple organs are also demarcated by its membranes. For example, it has been hypothesized that about 1.4 billion years ago, the mitochondria enters eukaryotic cells and begins a long-term symbiotic relationship. Possible, maybe it's because the mitochondria are once an independent organisms. They have their own DNA genome. RNA was once found to mostly serve as a messenger that carries instructions from DNA. However, in the last 30 years, researchers have identified multiple types of RNAs. Among them, the most important are the long-coding RNAs, the types which is not involving the protein productions. The non-coding RNAs constitutes more than 90% of RNAs made from human genomes. More than 50,000 known non-coding RNA has been identified, but most of them remain largely understudied. Still, there are many non-coding RNA have been shown to play important roles in cell functions. Similarly, that mitochondrial DNA can also transcribe non-coding RNAs. After transcription, some of these non-coding RNAs can be transported into the cytoplasm, which can be interfered by the conventional RNase technologies. However, some of them may stay within the mitochondria. As the mitochondria are the negative charge and have double membranes, it serves as a barrier for the RNase interference. Due to the efficient mitochondrial-specific non-coding RNA delivery systems is lacking, the functions of mitochondrial-located non-coding RNAs are largely unknown previously. The mitochondria are the centers of cell metabolisms. Metaphagia is an chronic inflammation caused by excessive nutrition. It's a hallmark of immune metabolic disease. The non-alcoholic fatty liver disease is a typical immune metabolic disease. It affects about 1/4 of the world's total populations. It is not only associated with other metabolic disease, such as diabetes and insulin resistance, it also associated with tumors. It not only associated with the hepatic tumors, interestingly, is also closely associated with breast cancers. And the NASH, non-alcoholic steatohepatitis, is a progressive type of the fatty liver disease. Therefore, under the stimulations of lipid overload, the metabolic disorders can be converted to the immune disorders, and then to the fibrosis, cirrhosis, and all the way to the tumors. The fibroblasts are the major source of collagens and many cytokines. And therefore, I collaborate with Dr. Gao Ziliang, who is a hepatologist. We isolate the human primary fibroblasts from the normal liver tissues and the liver tissue of NASH patients. By using the seahorse extracellular fast analysis, our data show that the NASH fibroblast exhibits elevated ECAS as well as ECA-OCAS ratios. It suggests that there's ATP production is shift from the oxidative phosphorylations to the glycolysis. Moreover, both sensory and mitochondrial roles increase in the NASH fibroblast. These data suggest that the lipid overload induced the metabolic reprogramming of the liver's fibroblast. As the circulites have been shown to play important role in multiple cell types, however, their roles in the fibroblast remain elusive. Therefore, we compare circular expression patterns between the NASH fibroblasts and the primary liver fibroblasts from the human patients. We assessed that 15th and 11th circular RNAs were consistently upregulated and downregulated in the NASH fibroblasts amongst the different patients. Interestingly, our data show that nearly 40% of the downregulated circular RNAs in NASH is transcribed by the mitochondrial genomics, despite less than 0.1% of the circular RNAs are divided from the mitochondrions. The fashion acids conferred 3 of these downregulated circular RNAs are located in the mitochondrions. Among them, one is overlapped with ND5 in the heavy strands of mitochondrial genomics, whereas the others located in the light strand. So how to investigate these mitochondrial located circular RNAs? I talked to my friend, Dr. Xiaoding Xu, who is a nanotech scientist. Together, we developed nano particle platforms to deliver the circular RNA into mitochondrions. We adopt mitochondrion targeting peptides, which consisted of mitochondrions targeted oriented and a membrane penetration sequence. The TPP is positively charged phosphorus atoms surrounded by several hydrophobic groups. Therefore, it can be attracted to the negatively charged mitochondrions with lipophilicity. Then we encapsulate these peptides with the circular RNA expression vectors in the nanoparticles. This nanoparticle contains a pH response polymers, and therefore these particles can be potentized under the acidic endosomal microenvironment, and then the circular RNA expression vectors are released in the cytoplasm and then delivered to mitochondrions through the specific peptide. Our data show that only around 10% of the GFP signal co-localize with the mitochondrions when the GFP expression vectors are transferred into the fibroblasts with the conventional lipid transfection reagents. By short comparisons, more than 90% of GFP signal co-localize with mitochondrions when the vector was delivered by incubating the fibroblast with the mitochondrial nanoparticles, suggesting the specific capacities of these mitochondrial nanoparticles to target mitochondrions. Amongst the downregulated circular RNAs, we found that most of the endogenous circular RNA scars located within the mitochondrions. Moreover, the northern blot analysis demonstrates that the circular RNA scarred mitochondrial nanoparticle treatment significantly increased the circular RNA scars, which is resistant to the RNase treatment. By comparisons, its lineage transcript, which is sensitive to the RNA treatment, remains unchanged. The mitochondrial membrane potential in NASH fibroblasts were much lower compared to the normal fibroblasts. The mitochondrial specific overexpression of circular RNA scars restores the depressed mitochondrial potentials and also inhibits the collagen contractions of the NASH fibroblasts. Then we try to explore the underlying mechanism of circular RNA scars. We identified putative open reading frames on the microRNA scars. So we insert a flat codon sequence immediately upstreams the stop codons of the putative ORF and finds no flat codons can be produced. So this data suggests that this circular RNA is non-coding RNAs. Moreover, we identified several microRNA binding sites in this circular RNA, but none of these microRNA contains more than 2 binding sites or are transcribed by the mitochondria's genomics. So this data excludes that the circular RNAs serve as the sponges for circular RNAs. Moreover, we do not assess appreciate change of its host genes by manipulating the circular RNA scars. Other and our species data show that the RNA binding protein plays important roles in the non-coding RNA functions. So we cross-link the protein and RNA by the treatment of formaldehyde, and then extract the RNA protein complex from the lysis mitochondrions. The lysis were incubated with the by telomerase, a DNA oligos pose, which targets the backslide junction of circular scars, and then the complex are pulled down by the magnetic agarose beams. The mass spectrometry identifies 8 binding proteins. Among them, the mitochondrion permeability transition port stood up as 2 of its regulators were identified. As ATP58 can bind to ATP5B, therefore we ask whether the circular RNA directly binds to ATP5B or 58. By using the in vitro RNA protein assay, our data show that the circular RNA scars specifically binds to ATP5B rather than 58, and can only retrieve the ATP58 in the presence of ATP5B. The ATP5B has been reported to anchors to the matrix facing inner membranes of the mitochondrions to investigate whether ATP5B and circular scars located in the same mitochondrion compartment. We use previous reports APEX targeting methods that identifies specific mitochondrion regions of RNA and proteins in the live cells. We transferred the normal fiber master with the APEX, which targets different mitochondrion compartments, and we tried these functions by the steroid rings. APEX is a peroxidase which is fused to the mitochondrion proteins of interest. After the administration of a substance, the bioctin phenols and the hydrogen peroxide to the live cells, it generates the bioctin phenol radicals and labels the nearby endogenous proteins. And then we pull down the proteins or RNA complex by the magnetic beams. Data suggests that both ATP5Bs and circular RNAs locate in the matrix rather than in the intermembrane space or the outer mitochondrial membrane. To investigate the clinical significance of circular RNA scar, we performed the FISH. Our data show that the circular RNA scar was located in the mitochondrions and predominantly expressed in the fibroblasts. Moreover, we found that circular scars exhibit a continued decline from normal liver tissues to simple steatosis all the way to NASH cirrhosis, suggests that it correlates with the non-fat progressions in patients. To evaluate whether the mitochondrial nanoparticles can be delivered in the mitochondrions in vivo, we label the circular RNA expression vector with immunofluorescence. Then we ingest the nanoparticles while the tail weighs into the mice. The nanoparticles exhibit the higher fluorescence compared to the naked vectors in most of the organs. Among them, the liver showed the highest mitochondrial nanoparticle accumulations. Moreover, we observed that most of the circular RNA co-localized with the mitochondrions in the mice administrated with these nanoparticles. More importantly, is the treatment of circularized scarred mitochondrial nanoparticles significantly reduce the fibrosis and the inflammatory cells infiltrations in the mice fed with high-fat diets. Moreover, it improved the tolerance of glucose in vivo. To make the long mechanistic story short, our data suggests that PGC1 alpha, a co-activation factors, mediates the expressions of mitochondrial scars in the which is transcribed from the mitochondrial genomics. This circulinate binds to the ATP5Bs on the membranes of mitochondrions and inhibits CYPD interactions with the MPTBs. During the lipid overload can induce the ER stress. The ER stress signals trapped can inhibit the expression of PGC1 alpha, and therefore the relief suppressions of interaction between CYPD and MPTBs by the circularized scars lead to the opening of MPTDs and the enhanced generations of mitochondrial roots, as well as its output into the cytoplasms. The cytosolic rods can serve as signaling proteins, which activates the fibroblasts and also drives the NASH progression. So the Great Wall is a monumental landmark in China and the longest structures ever built by human beings. It's a defensive system built across the borders to protect the peace and stability of the territories. Disney's Mulan is one of my favorite movies. On its opening field, the beacons is lit by brave soldiers when the bad guys invade. And it set up a chain of lighted beacon towers all the way to the capitals to alert the incoming invasions. This is a rapid border alert system adopted by the ancient Chinese more than 2,000 years ago. Both plasma membranes and organelle membranes are the borders of cells to maintain the environmental homeostasis within a certain boundary. Nucleic acids are very ancient signals. Our studies identifies a number of nucleic acid interactions signals on both the plasma membranes and the mitochondrial membranes. After detecting the DNA and the RNAs, these membranes can trigger a signal cascade to prepare the cells to better respond to the outer stimuli, which is quite similar to the beacons of the Great Walls. So I would like to thank my good members, collaborators, and the funders. When I get the invitations, I'm very excited. I talked to my friends. One of my friends told me that, hey, they do not invite you for your talks, they invite you for your Cambridge. I'm sorry for my absence because I don't have the privilege to study abroad. So I hope after this talk, you may remember some things beyond my Cambridge. Thank you very much, and questions are welcome.
Thank you, Shi-Cheng. So if people have questions, could they please put them in the Q&A so I can ask them? There's some questions from Yoon-Hee Yi, who asks, is CCDC25 specific to cancer cells? What happens if normal cells can express CCDC25?
This is an excellent question that we desperately try to address currently in collaboration with Dr. Song. We observed that the CD25s are overexpressed in the tumor cells, both in breast cancer cells and the colon cancer cells. Under the normal circumstances, the CD25s can also express in several normal tissues. First, it can express on the immune cells. In this, the previous reports have identified that the NETs can interact with the immune cells and mediate the disease progressions, multiple disease progressions such as atherosclerosis and autoimmune disease. Secondly, we found that it can be expressed in an endocrine, neuroendocrine cells in the lungs. We find it's very interesting. It's very interesting because, for example, when the patients get pulmonary infections, they can have sticky sputums. Why the sputum is sticky? It largely contributes that it contains lots of DNA components of NETs. So we know that the neuroendocrine systems response to this stimuli and lead to the cough or the hyporesponses of the respiratory tract. So maybe this CD25 also mediates this effect. The more interesting thing is we assert that an adhesion molecules of bacteria contain the exact the DNA sequence that we identify for the CD25 combines with the NET DNA. As we know that the NET has been firstly identified to capture the bacteria for the neutrophils. So whether this sequence may mediate the adhesion of the bacteria on the NET. So there's a lot of exciting questions that we're trying to explore.
Okay, so Dr. Yi also asked, does NET DNA physically directly interact with CCDC25, or is there some mediator? Is it a direct interaction?
First of all, is a direct interactions because we performed the MSI assays with the recombinant CD25 and the DNA. We find that the CD25 can form the complex with the DNA, especially the oxidized AODH enriched DNA. But whether the other components on the NET can enhance these interactions, my best guess is yes, but we don't know the detail yet.
So TREX1 doesn't digest the oxidized DNA, and the oxidized DNA binds to DC25. So there are two things that are working together. Is that correct?
We use TREX1 confirms that the DNA that we are isolated are indeed oxidized NET DNAs rather than the DNA fragments from the nucleus of the unstimulated neutrophils. TREX is a synthetic as a nuclease. In vivos, such as in mouse or in humans, whether this is mediated by TREX, we don't know. Maybe it's mediated by the other extranuclease. And we also suspect that other components on the NET, such as the granule proteins, such as the MPOs or the others granule proteins, may enhance the bindings of the DNAs and CD25s.
So would you predict that DNase the DNase is used as a drug in cystic fibrosis? Would you predict that DNase would reduce liver metastases?
Actually, this is, you mean whether the treatment of DNase can reduce the liver metastasis?
Yeah.
Yeah. Actually, this is not our discovery. It has been reported in cell models. And I think in certain circumstances, it may reduce the metastasis, depending on how much these models depends on the neck.
So is there a difference, if you look at breast cancers that metastasize to the liver, is there a difference in CCDC25?
Yeah. Yeah, we have been shown that both the selected the liver's preferential metastasis, the cell lines through the serous mouse injections and the human samples, the case with higher instance of liver metastasis shows elevated level of CCD25. Yes, we have shown that.
But what about in colon cancer, which metastasize to the liver more than other cancers? Is there any?
Yes, we have also had similar observations. We have two different cohorts, both the breast cancer cohort and the colon cancer cohort.
Oh, yeah. Okay. Well, I think our time is up. Thank you so much, Shi-Cheng, for your seminar. And we hope to see everyone soon at the next Connect Science seminar. Thank you.
Thank you for having me. Good day.